Are we still waiting? Time to start now.
Yeah, I think we're going to start today's session.
Okay, go ahead.
Okay. So hi everyone, I'm Karen, a volunteer of CAST Greater New York chapter. The COVID-19 pandemic has galvanized scientists and professionals across the globe to work this event. Our speakers will ask questions following an interactive. We will try our best to address them in the Q&A session. So today's webinar will be moderated by CAST-GNY Vice President Jia Cheng. She is also a researcher at Amsterdam Medical Center. Let's welcome Jia.
Thanks, Karen. Welcome, everyone. I'm Jia Cheng, the Vice President of Chinese Association for Science and Technology, Greater New York chapter. I will be the moderator of today's virtual event. I'm sure it's going to be a very insightful lecture. First, please allow me to invite our President Tao Tao to introduce the history and the mission of our association.
Yeah, good morning. Thanks, Joy. My name is Tao Tao. I'm the president of Chinese Association for Science and Technology, namely CAST-USA GNY New York chapter. Today I'm very excited to see so many distinguished speakers come to join this remarkable scientific education event. Before start, let me take 1 minute to give a very brief introduction of our organization. And Chinese Association for Science and Technology was founded in 1992 in New York, and we have total 16 established chapters across US and 8 to be built chapter. We have 12 liaison offices in China. Our mission is to promote science and technology exchange and education for both American and Chinese communities. We have many distinguished industrial and academia members, and we host various events, regular events, including like technical forums, entrepreneurship workshops, and startup contests. We have our annual conference coming in October this year. And I hope we will see each other again. Thank you so much for joining this event today. Joy? Joy, we cannot hear you.
Thanks, Tao. So we at the Greater New York chapter are committed to educating the public, keeping our audience stay informed about medical research and drug trials for COVID-19. Today, we will have the privilege of getting to talk to 2 distinguished speakers about all the big events, all the things that are happening on the front when it comes to the coronavirus. Joining us right now is Dr. William Hasseltine. He is former Harvard medical professor. He's also a very prestigious pioneer and contributor, founded more than a dozen biotechnology companies. And he is the chair and president of Access Health International. Dr. Hasseltine, thank you very much for accepting my invitation in this time of COVID-19, in this time of crisis of COVID-19.
You're very welcome, thank you.
And we have another special guest, Dr. Yaning Wang from the Food and Drug Administration. Dr. Wang, thank you for joining us. Welcome.
Thank you. Thank you for inviting me.
We're going to hand over to Dr. Wang shortly. But to introduce Dr. Wang, I'm privileged to have my co-moderator, Dr. Hua Lu from Tulane University in New Orleans. He is professor and the chair of the Department of Biochemistry and Molecular Biology. Dr. Lu, please.
Hello, hi everyone, welcome. It is my great honor and delighted to be a co-host with Dr. Jia Chen, and I will be happy to facilitate this and try to do my job. And first, let me introduce our first speaker, Dr. Yaning Wang. And Dr. Wang actually graduated originally from Peking University School of Medicine. I think it's a School of Pharmacy in 1996, and received his master's degree from National Doping Control Center before he went to University of Florida to pursue his PhD and the master's degree, and received degree in 2003. Then he started his job at FDA, and right now he's a director of division of pharmacometric in the Office of Clinical Pharmacology, and he's actually overwatching the review, policy development, research project, and cover the all kinds of disease. And this is certainly part of the area that he's covering. So let's welcome Dr. Wang to give us about 30 minutes of talk, and go ahead if you're ready.
Sure, thank you. Let me share my screen. Where is the share screen button? Can you guys see my screen now? Okay, good.
Hold on, I wish everyone else mute your audio.
Okay, first of all, thank you, Dr. Lu, for the introduction. And also, I would like to thank the organizer for inviting me to this very timely discussion. Today, I would like to share some work we recently published about hydroxychloroquine. As you can see from the title, we try to explain how to connect the hydroxychloroquine in vitro antiviral activity to the in vivo concentration for the prediction of the antiviral effect. I have to say today's presentation only represents my own opinion and should not be interpreted as FDA view or policy. Our work was recently published in the Clinical Infectious Disease on May 21st. As you can see, this is teamwork. I have multiple colleagues to work with me on this paper. And the bottom line of this paper is in order to correctly translate the in vitro antiviral activity of hydroxychloroquine or chloroquine, we have to link or we have to compare the right in vivo concentration to this EC50 or EC90 values that is typically reported as almost like a potency value for the drug's antiviral effect. The right concentration is the in vivo free extracellular tissue concentration, which is similar to the free plasma concentration. So I want you to remember that as the bottom line. So before we go to the details, I want to go over some of the basic facts about hydroxychloroquine. And I'm using hydroxychloroquine as an example, even though all the relevant discussion will also apply to chloroquine. So if you open the label for hydroxychloroquine, we know this is an old drug. It was already proved to treat malaria and RA and lupus. And if you look at the mechanism of action section in the label, you will see for malaria, we say, well, the exact mechanism or precise mechanism is not known, but we did propose some hypothetical mechanism. It says hydroxychloroquine or like chloroquine is a weak base. The compound is a base compound and may exert its effect by concentrating in the acid vesicles of the parasite. So this is a proposed possible mechanism of action. But for the RA or the lupus, where the drug is supposed to suppress the immune system, the current label basically says we don't know how the drug works. The mechanism is unknown. And another important issue is the dose, because by so far you must have heard a lot about chloroquine, hydroxychloroquine from the news or from TVs. And the one important issue is the dose. Again, we start with the approved dose for the approved indication. So if you look at the 3 major indications for malaria, again, it's further separated into prophylaxis and treatment. And prophylaxis is 400 milligram once a week. And then for the treatment, it's higher dose. That is, the first day you gave 800, then 6 hours later, you gave 400. So on the first day, you gave in total 1200 milligram. The next day, you gave 400, and the next day, you give another 400. So for lupus the indication, the approved dose is different. As you can see, the recommended adult dose is a range, is 200 to 400 milligram dose. Again, it always converts to the base because there is a conversion of the molecular weight, because the final drug is the salt, it's not the pure base. And this dose should be administered either as a once-day regimen or divided into 2 doses. For example, if it's 400 daily dose, it can be divided into 200 BID or twice a day. And then in this dosing recommendation, actually specifically it mentioned one toxicity, that's the incidence of retinopathy. And it says this is dose related. If the dose is too high, this toxicity will happen. And for the RA, again, the dose is different from the others. For example, again, for adult dose, the initial dose is 400 to 600 milligram daily, either as once a day or twice a day. And once it reaches the so-called good response, when the good response is obtained, then dose can be reduced to half of the initial dose. And again, it highlighted that do not exceed this highest dose, then the safety event will happen. So this is all the public information in the current hydroxy approved label. Then in addition to the efficacy, I also want to highlight the known toxicity or the safety warnings in the product label for hydroxychloroquine. And you can find similar information from the chloroquine label as well. The top 2 is, the first one is ocular. That's what the other 2 warnings were already talking about. This is the irreversible retinal damage that has been observed in some patients who had received hydroxy. And the second one, again, everyone must have heard it from the news, is the cardiovascular effect, or we call it QT prolongation, that could lead to arrhythmia or sudden death. So again, in this one, in the label, it says post-marketing cases of life-threatening and fatal cardiomyopathy have been reported with use of the hydroxychloroquine, as well as with the use of chloroquine. And there are more toxicity, like worsening of psoriasis or porphyria, or proximal myopathy or neuropathy, and a neuropsychiatric event, even including suicidality. And the last one, again, these are all the highlighted safety events in the product label, even though there's another long list of adverse events. So these are the most serious events. And the last one is hypoglycemia. So keep this safety event list in your mind when we're talking about risk benefit for hydroxychloroquine. And then, if you pay attention to the news, we have a series of FDA announcements. I guess we can start with the announcement of the emergency use authorization for chloroquine and hydroxychloroquine on March 28th. And then, of course, later on, we FDA also published this safety communication, cautions against use outside the hospital setting or clinical trial due to the risk of heart arrhythmia problem. This is the QT prolongation issue. So the emergency use authorization only allowed the drug to be used within the hospital setting or a clinical trial. Then, of course, our paper was published on May 21st. Then shortly after that, FDA, we published a short blog, or we call it a CEDAR Spotlight, again, summarizing what's in the paper and put it on the website on June 3rd. Then I guess, then you all know on June 15th, FDA revoked the EUA for both chloroquine and hydroxychloroquine and provided the justification in a separate letter. So this is the timeline of all the major events related to the hydroxychloroquine use. Then I will get into the sort of the scientific details of this drug. I want to put the hydroxychloroquine and the chloroquine side by side to start this conversation. As you can see, the molecule structure between these two products is very similar, except there's a hydroxy group at this position. In fact, if you again read the label, the share similar kind of pharmacokinetics, even though only the chloroquine label highlighted that the drug is deposited in the tissue in a considerable amount, and specifically giving the number, like in animals, these tissue concentrations were measured. It showed that 200 to 700 higher concentration in the tissue were observed than the plasma, and depending on which tissue we're talking about. And in the hydroxychloroquine label, it didn't say that, but it's, I guess, expressed in a different way. It says the absorption half-life is 3 to 4 hours, and the terminal half-life, that's how fast the drug gets out of the body, is very long. It's 40 to 50 days. And it further explained that the long elimination or terminal half-life can be attributed to the extensive tissue uptake or the accumulation rather than through the decreased excretion. So both of these two sentences are highlighting the drug can get into the tissue extensively, therefore leading to a much higher tissue concentrations than the plasma concentration. But we'll understand what that means in the next few slides. So after the coronavirus pandemic started, a few groups in China studied the in vitro antiviral activity of multiple compounds, including chloroquine and hydroxychloroquine. Here I'm citing two of them. On the left, this is a paper from Liu et al., and this paper actually first came out. The paper studied both the chloroquine and hydroxychloroquine under the different conditions of MOI, means a multiplicity of infection, which means the relative amount of virus that was added to the in vitro cell system. For example, if the MOI is equal to 0.01, that means if you have 100 cells to be infected in the in vitro system, you add one virus. So it's 1% of the total cell. So as you can see, when you increase the MOI to higher and higher number, I don't know whether you can see this, the EC50, or the half the concentration of the drug that can suppress half of the maximum inhibition of the virus, that's corresponding to EC50 or EC90. I know the audience is a broader audience than the general scientists in the pharmaceutical field. That's why I may have to explain these curves a little bit more. On the x-axis, that's the concentration, increasing concentration of the drug, either hydroxy, HCQ, or CQ chloroquine. On the y-axis, that's the percentage of viral growth inhibition at a different concentration level of the drug. As you can see intuitively, when you don't have any drug or very low concentration of drug, there's 0% of inhibition. So to do this kind of experiment, you have a control arm. That is, you don't put any drug in the cell culture media and let the virus freely grow within the cell you put there. So that's the reference. So you use that as a reference to calculate, especially when you have high enough concentration of either hydroxychloroquine or chloroquine. As you can see, you can almost reach 100% inhibition of the virus growth, meaning it doesn't matter what the mechanism that is, if you give enough drug in this in vitro system, meaning outside the human body in a test tube, if you give enough drug, either CQ or HCQ, it can inhibit the growth of the virus in this experiment. The value here called EC50, that corresponding when you reach 50% of inhibition, what is the concentration on the x-axis? Of course, the smaller this number is, the more potent the drug is. And one thing you can observe is when you add more and more virus at the beginning of the experiment, this is almost like 80% of the virus relative to your host cell. This number becomes larger and larger. That means it's getting harder and harder to stop the virus from growing, or you need more and more drugs to reach 50% of the inhibition, like the same amount of inhibition. So this experiment actually is from the Liu et al. And on the right side, we have another independent experiment done by Yao et al. This was later published. Again, the similar plot was generated, one for chloroquine, one for hydroxychloroquine. Here there are two curves. The difference between the two curves is 24 hours or 48 hours incubation with the drug. And for this experiment, the incubation of the drug together with the virus and the cell is always 48 hours. But in this experiment, they also explored 24 hours. But the bottom line is, again, the highlighted or summarized the potency of the drug against this virus based on this EC50. So again, when we list these numbers, as I mentioned earlier, for both drugs, increasing MOI will lead to a higher concentration, meaning you need more drug to inhibit the virus growth. But the general trend is hydroxychloroquine has a higher number, meaning less potent than the chloroquine in this experiment. But in this Yao et al. Paper, they got an opposite result. That is, hydroxy had a smaller number, meaning it's a more potent drug than chloroquine. But there are more papers like this. After these two papers were published, more papers came out with similar experiment, and there are trial-to-trial variability on this number. Therefore, you should consider the range of EC50 or sensitivity of the drug against the coronavirus. One thing I want to highlight on this one is the experiment on the left is actually conducted under a so-called prophylaxis condition. That is, they gave the drug first to the cell, the host cell, so that the cells are protected by the drug before the virus was introduced into the system. And on the right side, it's treatment, meaning the virus was first introduced into the cell to infect the cell. After that, the drug was added to see whether the drug can inhibit or slow down the virus growth. So there's a key difference here. That's why I picked these two just to highlight both the prevention or prophylaxis cases and the treatment cases are covered. But surprisingly, typically intuitively, you would think the prophylaxis should have, you need less drug to inhibit. But between these two experiments, you can see the prophylaxis case actually need more drugs than the treatment. In fact, I would say, even though it's not on this slide, if you go to the paper of the Yao et al., they also did prophylaxis experiment. And somehow they also ended up with higher EC50 for the prophylaxis than the treatment. Then the Yao et al. Paper moved on to simulate or predict under the current safe dosing regimen, what would be the in vivo, the concentration inside the human body, so that they can compare that concentration to see whether that's high enough to inhibit the virus. So on the left, that's the predicted concentration in the plasma, in the blood, over time, under various dosing regimens. I will have a table to show the detailed regimen under this A, B, C, D, E, F. But here I just select a few to highlight, its relevance to the EUA, the FDA initially approved emergency use authorization regimen. So initially FDA approved a regimen that is 800 milligram on day one, followed by 400 once a day for the remaining either 5 or 9 days. So it's a 10-day regimen. And then, of course, in this paper, that exact regimen was not sort of used, but there are some regimen that's very close. For example, regimen B is day 1. This is almost like the, I would say the malaria treatment regimen. First day, you gave 800 plus 400. After that, you gave 400 every day for the remaining 10 days. Then there's another regimen that is on first day, the 800 milligram was split into 400 two times for the first day, and followed by, again, the 400 was split into 2 times, 200 twice a day for the remaining 5 days. But irrespective of the regimen, as you can see, the predict concentration, especially at the steady state, they're all pretty close. And of course, this pink line is for chloroquine as a reference. The regimen is 500 milligram twice a day for 10 days. And the remaining, lines are for hydroxychloroquine under different dosing regimen. On the right side is the predicted lung concentration, the tissue concentration, again, under all the different regimens for both the CQ and HCQ. As you can see, the y-axis is on a log scale. And roughly, the steady state, we call the steady state or the stable concentration for hydroxy is about 400-fold higher in the predicted lung tissue than the plasma concentration. And this is the key, because this actually came probably directly from the label, where remember you have the 200 to 700-fold higher. This number roughly came from that animal study. And then the next question is, when you compare this tissue, the lung concentration, with the EC50 that was from the in vitro antiviral experiment, the author came up with this table in the publication that showed for either the chloroquine or the different regimens of hydroxychloroquine, the ratio of the free lung concentration over the EC50, that's the potency number from the antiviral experiment, they're well above 1, or it could reach as high as like 169, meaning the concentration looks very high compared to the in vitro potency number, suggesting either chloroquine or hydroxychloroquine under the current proved safe regimen, they should be very efficacious in terms of inhibiting the virus based on this in vitro, in vivo correlation. And this is not the only paper. I would highlight, after that publication, there are many more papers coming out with similar conclusion. For example, here, this is the Liu paper. That's from the one of the in vitro experiment. They also highlighted, since the tissue concentration is so much higher than the plasma, therefore compared to what they measured, the EC50, even though their number is higher, remember it's like 4 point some micromolar, the in vivo tissue concentration should be well above that number. And in this paper, again, they quoted the same sort of high fold tissue concentration, again suggesting it's very likely the current regimen will easily achieve the antiviral concentration. And this one actually went even further. They even conclude that it's possible even a low dose, like the first dose 400 followed by 200 once a day, should also be enough, if the tissue concentration is so high. Then this one also, similarly, they all believe, the high tissue concentration should easily achieve the in vitro potency number. I think this one is probably the most extreme one. That is, they think the simulated unbound lung concentration was above the EC50, and suggesting even a subtherapeutic dose of 10 milligram dose is probably enough. And again, this is another paper basically saying the same thing. And this, again, there are so many, I did not collect all of them. This is probably the most, actually not the most recent one, the most recent one before we publish our paper. So this one, again, compared the in vivo concentration against the EC50 of many different compounds and calculate this concentration over EC50 ratio and ranked them. As you can see, hydroxy One meaning because there are different EC50 values reported from different papers. That's why you have hydroxy 1, hydroxy 2. That's like using different EC50 values from different papers. Hydroxy 1 ranked number 1 with a ratio that's close to 1,000, meaning the predicted maximum concentration in the lung is 1,000-fold higher than the EC50 from the in vitro, suggesting this drug should be very potent. Well, enough with all the published paper with that conclusion. If one thing I want you to remember from this talk is this picture. In order to correctly link the in vivo concentration to the in vitro EC50, we first have to understand how the in vitro antiviral experiment was conducted. On the top, I guess, part of the picture, we use this cartoon to roughly explain how the experiment, the in vitro experiment was conducted. Like I said, it's in a cell culture. You first put a lot of cells. It's almost mimicking, mimics the human host cell. Then you add virus, whether it's before you protect the drug with the drug first, or you introduce the virus first, then add drug to treat. Then in the cell culture media, you add drug. And the concentration, remember in the XY plot, the x-axis, the drug concentration from low to high is the concentration in the cell culture media. And remember this, well, one thing I highlighted earlier is both hydroxychloroquine and chloroquine, they're base. They have these three nitrogen molecules in the molecular structure that led to their pKa above 7. That's why once they get into this highly acidic intracellular vesicles, such as lysosome or endosome, any intracellular vesicles that have very acidic environment, this neutral hydroxychloroquine or chloroquine will be immediately ionized under this acid condition and pretty much stuck inside this lysosome. In fact, this is the exact mechanism why there's such a high tissue concentration for both compounds. Is it because in the blood, they're in a neutralized format and they can enter the cell. This is the human host cell or the cell they put in the in vitro system. But inside the cell, there are multiple intracellular vesicles that have very acidic environment. And there's a reason for that. Once these drugs enter this compartment, they are highly ionized. That's why they're stuck. They cannot get out. They cannot move freely across this membrane. And this is the exact reason why we have 200, 700-fold higher tissue concentration compared to the plasma concentration. But remember, the drug concentration, all the paper reported to reflect the potency of the drug is not the intracellular concentration, is the extracellular concentration in the cell culture media. In other words, if, for example, we take the EC50, let's round it up to 1 micromolar, meaning if you have 1 micromolar of drug in this space, that can suppress 50% of viral growth. That means probably you have 1,000-fold higher concentration in the intracellular space to have that effect. Okay, with that, now you understand when we move to the human lung tissue, what is the corresponding cell culture in the tissue? That's corresponding to what we call it lung interstitial fluid, and what corresponds to this kind of intracellular compartment. Again, this is the human host cell. The only reason or the main reason for the high tissue concentration is all the drugs are stuck in the intracellular compartment. And this is, of course, the corresponding space that's coming from the human lung or any tissue. So in other words, in order to have an apple-to-apple comparison, we need to compare the drug concentration in this space, the extracellular space, in order to compare it with the 1 micromolar or 4 micromolar EC50 calculated from the in vitro experiment. But as you can see, all the papers I just presented, they compared the concentration pretty much inside the lysosome or endosome, all the intracellular concentration, which is, of course, 100-fold higher than the extracellular concentration, suggesting, this is much higher than this, but in fact, the intracellular concentration here is probably similar to here or even lower if the accumulation in the in vitro system is higher. So with that, we recalculated the right concentration over the EC50. By the way, in the antiviral drug development, EC50 is a very low bar. Most of the sponsors to develop antiviral drugs, they will not target just EC50. They will target EC90. That is the concentration that will suppress at least 90% of the virus growth. Anyway, even with this EC50, you remember in the original table, they're very large, well above 1 or 50 or 100. But once you use the right concentration from the extracellular or the interstitial fluid from the lung or any tissue, depending on which EC50 value we use, remember one paper, the Yale paper, reported a very low number. This is actually the lowest EC50 reported in all the literature. And then of course, the other one is 4.5 from the other in vitro. They are well below 1. In fact, these are very small numbers. In fact, even if you plug into the EC90, which is much higher than these numbers, the number will be even lower. You can roughly just divide this number by 9. Then you get the ratio for EC90. Anyway, what does that mean? If you put that number back on the original plot where they showed increasing concentration of hydroxychloroquine will have more antiviral effect, the red range is the right concentration, in vivo concentration, again, inside the human body, the correct concentration. As you can see, this is almost close to placebo. You're not suppressing any virus growth. Similarly, this is, of course, we use the 4.51, the Liu paper as the exposure response curve. And of course, most of the published papers, they thought this very high concentration, basically almost 100-fold higher concentration from the tissue was the driving force, which is wrong, as I explained earlier. And similarly, if you put the two concentration range on the other plot where you have different EC50 depending on the 24 hour, 48 hours, again, this is the sort of the in vivo drug concentration corresponding to how much virus you can suppress. In other words, the conclusion is the in vivo concentration under the currently approved safe dose is too low to show any potential antiviral effect. Now, there are multiple papers, the authors were trying to use not those predicted concentration, but measured concentration in their own experiment. Many doctors or hospitals, they try to compare their observed concentration in the experiment with this EC50, especially this number, because this is the lowest one, easiest one to achieve. And it turned out there are so many traps for hydroxychloroquine or chloroquine because of their unique chemistry or this in vivo distribution feature. It turned out that because of they are highly accumulated in those intracellular components, even in the blood, they are highly accumulated or concentrated in those thrombocytes or granulocytes. Because of this, if you measure different concentration, meaning if you measure the whole blood concentration or even serum concentration versus plasma concentration, you will end up with totally different numbers. Because when you measure them, for example, you measure whole blood concentration, then you include everything that included all inside those cells. That's why the blood concentration over plasma concentration is about 7-fold. Here I have to specifically thank all these authors and all these groups. They did all these experiments way back then in the 80s. And in fact, a lot of excellent research work was done back then that helped us to quickly reach our conclusion with a lot of confidence. Without those people's work, we would not have been so confident about the final conclusion. Anyway, these people did extensive research to figure out where the drug got stuck in and why even the serum concentration and plasma concentration is different. You see, it's a 2-fold difference. It's because when you put the blood there to wait for it to be like clotted to collect the serum. Even during that process, those cumulative drug inside those cells, they were actually released back into the water part of the drug. That's why if you wait for too long, what you measure in the serum is not the actual concentration in the blood part of the drug. And even in the plasma concentration, the drugs are not 100% free, and half of them is bound to some protein. Therefore, there's another factor you need to convert. Therefore, there are so many traps that some of the scientists who wrote the paper recently did not pay attention to this. And they say, oh, look at my blood concentration. That's very high, higher than any of the EC50 reported. So it must be working. So in fact, this is one of the papers. You may argue, the previous concentration is, let's say, the mean concentration or the median concentration. There must be between subject variability. Some individual may have very high concentration. So this is the one paper that showed the individual blood concentration, the whole blood concentration, even if you pick the highest one, and of course this is milligram per ml, you convert that to the micromolar, and then you need to remember, do the blood to plasma, then eventually to free plasma. You end up with a much smaller number. This is even below the lowest EC50. And similarly, if you pick the paper, actually this is the first paper that triggered all the interest because of the, I guess, a flawed design to compare the viral load. And there were many literature editors or criticism on this paper, but to question their viral load data. But here they have the plasma.
Sorry to interrupt, Dr. Wang.
Yeah.
Because we have limited time, so maybe it's the time for you to wrap up a little bit.
Yeah. I'm almost there. So this one, again, if you take the observed concentration, take the median, of course, this one is not plasma, it's serum. So again, once you convert to the right concentration, again, it's well below the EC50. So there's all these steps that need to be carefully monitored. In fact, one of the simplest steps, that is how much centrifuge force you apply when you process the blood in order to get the right plasma concentration. Again, these people, the earlier scientists figured that out. They said you have to use at least 1,000. In fact, one paper only used 250, and they thought they measured the plasma concentration. It turned out their measured plasma concentration is as high as the blood concentration. And they even developed a PK model, which was even used by some scientists recently to predict the wrong concentration. And after all this criticism, I have to highlight one paper that eventually came out on June 5th. And this is a group from Duke University. And this group was trying to reproduce the Yao et al. Conclusion for the pediatric patients. They actually figured out the same problem. As you can see, they also realized the unbound hydroxychloroquine in the lung interstitial fluid concentration is much lower than the EC50. So this is so far the only paper that reached the right conclusion. Anyway, in summary, I want to highlight or re-emphasize the in vitro EC50, they represent the extracellular concentration in the cell culture, should be compared with the in vivo extracellular tissue concentration, which should be similar to the free drug concentration in the plasma. And of course, there's an assumption here, that is the accumulation ratio. If the accumulation ratio is similar between the in vitro and in vivo system, then the current drug, current safe and effective dose, is not going to reach a high enough in vivo concentration to have any antiviral effect. Again, in the paper, we highlighted we're only focused on the antiviral effect, but remember the drug was also approved for those RA or rheumatology indications, suggesting the drug has some immunomodulatory effect. So we leave that open. But even that, if you look at the recent well-designed randomized trial from Recovery or the WHO, those well-designed trials failed to find any effect in the studied population for hydroxychloroquine. So finally, I would like to thank my colleagues on the right. They are the co-authors who worked with me to publish the paper. And I would also like to thank all the senior management leaders from FDA, specifically within CEDAR. They supported us and guided us and helped us to eventually deliver an objective and scientific-based message to the public health. Finally, thank you very much for listening, and I'm sorry I probably went over the time.
Thank you. Thank you. Let's thank Dr. Wang to give a very great outline and overview of the effect clinical study on the hydroxychloroquine. So if Dr. Hasseltine sign back on, we could start a second section. Is that Dr. Hasseltine's on? Yeah. Yes. Welcome back. I know you're busy. And at this section, we are starting a second section in a different way and basically in a conversation with Dr. William Hasseltine. And this section will be, co-host with Dr. Jiayu Chen, mostly or mediated by Jiayu Chen, and occasionally I will chip in a little bit as well. So let me first give you some, background about Dr. I know he's well known to most of us, if not every one of us, but I still, it's a politeness, I need to give you audience some background. Dr. William Hasseltine actually graduated with a bachelor degree in chemistry at Berkeley, UC Berkeley, about 40 or so years ago, and 60 or so years ago. And then he went to Harvard, pursue his PhD in biophysics. And actually under the two Nobel laureate, James Watson and Gilbert Waters. And after that, he pursued his short-term postdoc study with David Baltimore. And that's probably then is why he started work on the HIV later on. And he started his career at Harvard University School of Medicine at the new institute called Dana-Farber Cancer Institute. I would say just started not too long ago before he joined that institute as a faculty. And he go all the way to raise up to the distinguished professor over there at Harvard until he showed other interest in start a company. And his major contribution is to, at least I believe, 3 areas. One of the areas is HIV. He's one of the person developed the first, idea and work on the HIV and grab a lot of fund from Congress. And also he made a tremendous contribution to cancer research and tumor biology. And also he's one of the founding member for the Human Genome Institute, and that's co-funded by the private sector as well as federal fund. And later on, he's very interested in donate his time to the public health, to the philanthropy. So he funded 2 private charitable foundation. One is called Foundation of Science and the Art. The other one is currently he serves as a president chair, Access Health International. So I will not waste more time and let this section go and with Dr. William Hasseltine. So to start this sections, let me open a first question, a simple question with Dr. Hasseltine. So during this crazy COVID-19, what do you think is the most promising way to handle this guy? Is a therapeutic, like Dr. Wang just presented, pottery, or vaccine? Therapeutic versus vaccine, which one you think the most important or equally important?
Well, first of all, thank you for the opportunity to speak to you today. And it's a extremely interesting topic, what's happening to COVID around the world. I regret to say that I found the previous presentation uninformative. We know that the drug is dangerous. We know that it doesn't work. It was a long explanation of a very sad chapter in this event where drugs have become politicized rather than understanding how they work in patients. We may have a theory of how it works. There's a serious flaw in all the work on hydroxychloroquine, and that is the virus doesn't enter the human body the way it enters cells in culture. And therefore, the hope that hydroxychloroquine would work was never a very good hope. And it proved to be disastrous and maybe fatal in a number of cases. It's going to go down in history as one of the FDA's major mistakes and open it to criticism that it was unduly influenced by politics. And that's happened not only in the United States, it's happened around the world. So we were witness and we heard the justification of why the FDA might have thought it was a good idea, but what it turned out to be was a very bad idea. And I'm very pleased to see that they reversed their recommendation. But unfortunately, that reversal of the information is not widely understood, and people are still using hydroxychloroquine around the world in one way or another, possibly with additional harm. So I personally was distressed by the previous presentation, very distressed, I have to say. And I apologize to you, doctor, who did your best job at presenting the data and the justification for something that turned out to be a serious mistake. The question you asked actually has a different answer from the one you might expect. This virus is possible to control without a drug or without a vaccine. All you have to do is look at what China has done. Look at what South Korea has done. Look at what Australia and New Zealand has done. You don't need a vaccine or you don't need a drug to control this epidemic. If you have the three key ingredients, and you probably only need two of those, but if you have these three key ingredients, humanity is capable of stopping this infection, stamping it out across even when the infection is already taken root, as it did in Wuhan. You need leadership from the politicians at all levels. And if the leaders are in the way, you remove them, as they did in Wuhan. You need leadership which is capable of communicating to people in clear language, in consistent messaging, leadership that is credible and compassionate. I would say China may have been short on compassion, but was pretty good on the others. The second thing you need is an organization. You need a national public health service that when people say jump, they jump right down to the local level. You cannot manage a national crisis through regional initiatives, as we in the United States have tried to do. And thirdly, you need a population that is got a sense of social solidarity. If you have a bunch of individuals that believe that you are infringing somebody's right to ask them not to harm others, you have a difficult situation. And unfortunately, the US is suffering from three deficits. You can look at Europe, you can look at other countries that have a modest response to this disease. They had a big epidemic. They're controlling it, not perfectly. Their people aren't behaving as they should in some ways. You have several hundred cases in some countries. If you look at Europe altogether, there's a continual infection going on. It has not been fully controlled, so that's a danger. But if you look at the United States, we're in the middle of a real disaster because of the failures of those three organizations. Two of them are longstanding, have nothing to do with current politics. The one of them does have something to do with current politics. It's a unfortunate situation. Now, when it comes to drugs and vaccines, so I will get to your question. I would say there's a 50-50 chance, in my view, that we are going to have a vaccine which is both safe and effective in the sense that it at least controls the symptoms of infection if it doesn't control infection altogether. But there again, I'm very worried about what is happening at the overall level. I'm very worried, for example, to see what's happening in China with the use of an unproven vaccine for the military. The people who are using that vaccine cannot know that it's effective, and they cannot know that it's safe. It may be effective, it may be safe, but you cannot know that from the data on which they are acting. I am afraid that the United States may go down the same route. Now, we have the FDA that says the vaccine must be effective in reducing disease in 50%, at least by 50%. Now, exactly what that means is a little uncertain. We need transparency, so we know exactly what that means. And of course, they say they won't release the drug or the vaccine unless it's shown to be safe. But we don't have a clear indication from them of what they mean by safe. Safe for 100,000 people? Safe for 100 million people? Safe for 1 billion people? Anybody who looks at drug safety knows there's a very big difference in what you require for a safety profile for a drug you're going to give to several billion people from a drug you're going to give to several thousand people. There's an enormous difference in the amount of safety studies that you have to do. It is not possible, in my view, looking at all the drugs that I've developed and have been approved, and vaccines, to assure that a virus will be safe in the amount of time we have. You cannot do a 1-year safety study in 6 months. You cannot test hundreds of thousands of people in the time we have to know that the vaccine is safe. Just think about a 1 in 10,000 serious event would be when you give a drug or a vaccine to 1 billion people. It's a difficult problem. I've said this publicly many times. We have not yet seen from the US FDA a coherent response. We haven't seen their action plan, and we haven't seen that from the National Institutes of Health, which is running these programs. So if we are to control this epidemic with a vaccine, the first issue is safety. The second issue is efficacy. Now, all of the data that I have seen suggests that none of the vaccines that are currently being tried can protect from primary infection. If you look at the closest model we have, which is primates, none of the vaccines for SARS, for MERS, and for this virus protect from nasopharyngeal infection. And in fact, when you look at the amount of virus, at least as is published, in the nasopharyngeal cavity of the monkeys that were vaccinated and then challenged, they have just as much virus in the nasopharyngeal cavities as those that were never vaccinated. Now, that isn't very quantitative and the data is poorly presented, but nonetheless, that is the statement. Therefore, the fallback position is we're not going to stop infection, but we may stop and modify the disease course. Very hard to know because we don't have primate models that get disease. And as we have learned, this virus is protean in its ability to cause disease. Every day goes by, we learn more about the diseases that this virus causes. Will a vaccine that doesn't stop infection stop all of the symptoms? Will it even stop even the most lethal of the symptoms, which may not be the lung? It may be blood clots. It may be some other aspect of this. Those are serious questions that need to be addressed. I'm not saying it won't work. I'm saying we don't know whether it will work. And if what happened with hydroxychloroquine happens with a vaccine, which I believe is highly likely, we are likely to get emergency use authorization for a vaccine for which we are uncertain to its safety and uncertain to its efficacy. Let me go a little bit further than that. If you are going to get some of these vaccines to be potent enough to work in elder populations, which are resistant to vaccination a priori, you've got to use potent adjuvants. And those adjuvants themselves, as many of you know, pose serious risks. There was a type 3 adverse event in one of the Moderna trials with only 8 patients. And these were healthy young people. What will happen to a fragile 80-year-old given something that knocks a 30-year-old on his ass and puts him in the hospital 3 days later? Those are very serious questions, and they're inherent in the need to get potent adjuvant activity, not only for the vaccines, because some of these may need potent adjuvants, but because of the nature of the population which is being treated, i.e. elder patients. So those are serious questions that anybody should be asking about a vaccine. And I would call on the US FDA to be clear to the public on the criteria on which they would judge a vaccine to be suitable for administration to all Americans, young and old, with the usual exceptions for those people who are immunosuppressed for one reason or another. That's on the vaccine front. On the drug front, it's much more hopeful. As many of you know, there are drugs that were specifically being developed to stop SARS and MERS. These were based on the known activities of the polymerase, the known activities of the protease, the requirement for a helicase, and others. There are very nice reviews that show those drugs were very potent. Now, if you looked at those charts for hydroxychloroquine, even in the cell culture, they were way up there in the multi-nanomolar. There are now picomolar drugs, I believe, that people are developing. And so there are going to be a whole range of drugs, whether those are chemicals that interact with the protease, the helicase, the polymerase, or a variety of other targets. And I'm convinced the more we look, the more we'll find. We are all exhausting at this point the existing pharmacopoeia, and AZT has not appeared. However, in the wings are the drugs that were developed for MERS and for SARS. They're being updated. They're being modified slightly, so there's new patent life given to those drugs. And I think you'll see them coming to the fore reasonably quickly. If we look at the 2 drugs that have emergency use authorization, hydroxychloroquine and remdesivir, there's one enormous gap in the data, as far as I can see. And that is in human antiviral potency. You just don't see it. If you look, for example, at the paper published by Chinese scientists on the use of remdesivir in severely ill patients, they measured the virus in bronchial lavage and they saw no difference whatsoever. What use is an antiviral drug that doesn't drop the viral load? And why haven't we seen Gilead measure antiviral levels in people who are given the drug? You may have noticed that they recently claimed at a public meeting, the International AIDS Society, that there was an improved benefit in severely ill patients. They didn't do a controls trial. They had about 300 patients that they gave the drug to, and about 800 that they said real world analogs. What are those real world analogs? Without the published data, we don't know. They claimed some benefit, that there were more people survived in their 300 than in the 800, but without a real controlled trial, you really don't know. And when a controlled trial was done, you saw that there was no effect. The Chinese paper was extremely clear. No effect on disease, no effect on death, no effect on viral load. Where is the remdesivir viral load data? An antiviral drug that doesn't drop viral concentrations in patients is not an antiviral drug.
Doctor,
Simple.
thank you. Thank you for the long review about the therapeutic versus the vaccine. So I have, I wanted, through this, by listening to Dr. Wang's talk and listen to your conversation, I come up with, in terms of a drug discovery vaccine, I have two questions in my mind. One is, because based on what Dr. Wang, my own literature information, there's no mechanism, no one knows what this drug targeted for. And however, there's a potential effect show up with because in vitro doses and tissue doses. And based on this information, my question is that how, how you pick out when you FDA, this question I'll through to both of you, and the mechanism versus efficacies. Which one's more important? Because without any detailed mechanisms, when I try to involve this drug discovery, if we don't have a mechanism, don't have a specific target, then we presume FDA will not prove those. However, this drug quickly put into clinical because previously proved as a drug for the other diseases. And so my question is mechanism versus efficacies. That's number one. Number two question is that what do you think? Because based on your HIV experience, and we know that initially when we try to develop a drug against HIV, everyone target individual target. Later on was to find out the combination, so-called cocktail treatment is effective. And I have not yet seen, but I know this started to develop now, effective development of the combination of treatment, the antibody versus small molecule, or the vaccine versus combined with the therapeutic. So the second question, what do you think the combination of treatment against COVID-19? Through this, both of you, if you are able to, Dr. Wang's able to answer that as well. Thank you.
Would you like Dr. Wang to answer first? I'm perfectly happy to.
Go ahead. Go ahead.
Okay. In terms of the first question, if you have good evidence that whatever you're doing drops viral load and drops it quickly, I think you don't need to have the mechanism. If you had a plant, let's say you have an unusual chemical from a South Sea coral that drops viral load and it shows clinical efficacy, you don't need to know the mechanism. You'll be perfectly happy with that drug as long as it's safe. So that's the answer to the first one. Second question, with HIV, from the very beginning, we understood there would be viral resistance. AZT showed up very, very quickly. And in our studies of the viral genome, when we were one of the first to sequence it, we pointed out all the different targets. And because I was then chairman of a department of cancer pharmacology, I proposed that we use combination chemotherapy right away, and that we develop multiple drug targets. As you now know, there are 25 different drugs developed against 5 different targets. There are 2 or 3 other targets I think that are good. Let me say on Gilead's behalf that they just published one of the most beautiful papers I have ever read on drug discovery of a drug called GS-6207, which is a capsid drug. It interacts with a capsid at a very low concentration, 10 nanomolars. It is one of the most beautiful papers I have ever seen. And it drops viral load in patients as well. So it is a fantastic piece of work. And there the mechanism is extremely well known because you know at the molecular level exactly what contacts this drug makes and exactly how it interferes with the viral cellular protein interface, which is critical to establish infection. So you have all the criteria in place. Let's hope that we can do that for COVID. I am confident we are going to have drugs that are not only therapeutic, so if you're infected, you never get sick, but are prophylactic. And those won't all be antibodies either. In fact, right now for HIV, there are 2 drug candidates, one an integrase inhibitor and one a capsid inhibitor, both of which last with a single injection for many months and are very suitable in terms of their safety profile, as far as we can tell at this point, for prophylactic use for those people who are likely to be exposed. So I think in the context of the advances, we can think of monoclonal antibodies and antiviral drugs as a bridge to give us the time to develop a vaccine which may be safe and at least partially effective.
Thank you, Dr. Hasseltine. Dr. Wang?
Yeah, actually, I will have a very short answer. For the first question, no, you don't need to have a clear understanding of mechanism of action to prove a drug. In fact, if you look at the labels for a lot of the approved drugs under the mechanism of action, you will see it's unknown or we don't know. So that's not a requirement. As long as you show, for example, in the randomized phase 3 trial, the drug does improve the clinical endpoint. That's the key. Of course, knowing mechanism is always good because that increases the chance of success. Even knowing a lot of the mechanism, we still have a very low success rate overall. If you just blindly try anything without knowing the mechanism, the chance of success will be even lower. And again, Dr. Hasseltine, I would like to take some seconds to clarify. If I did not explain my presentation clearly so that let you make that comment, I'm sorry, because in fact, the whole point of my presentation is to explain why FDA reversed the initial decisions. Our analysis was cited as the #1 reason why FDA revoked the original EUA. So I hope I was trying to explain that clearly. Probably I didn't get that message clear to you. But my point is, because of this, therefore, we think, together with additional evidence, of course, that's why FDA changed the initial EUA and reversed it. I want to clarify that to you. Thank you.
How about your answer to the question about what do you favor, a vaccine or a drug?
Yeah, again, vaccine is now reviewed by or approved by our CBER. It's a different center. I work on in the CEDAR, the different center. But again, I noticed that FDA just published a very specific guidance for the vaccine development, what requirements we will need, and that's published on June 30th, very recently. And in terms of which one has a better chance, again, like you said, probably equal chance. And there are a lot of new technology these days in the vaccine field. We have to see whether that will eventually turn out to be efficacious. Again, I learned all this vaccine against coronavirus, including the flu, at the beginning of this pandemic. To be honest, the confidence level is pretty low. If you look at all the proved vaccine against the typical coronavirus, including the general flu, it's not that great. But I guess FDA laid out the sort of a minimal requirement. That's the 50% improvement in the guidance. So that kind of effect size, I guess, will ensure that only the very good and effective vaccine will be approved. On the drug side, I think we will not lower the standard for regular approval. For the EUA, is clearly a different standard. We have a different law that says as long as you think it's maybe effective, FDA can issue an emergency use authorization, but that's certainly not approved drug. I think FDA will stick with our normal standard to prove a safe and effective drug or vaccine. Thank you.
Thank you, Dr. Wang. Thank you, Dr. Hasseltine. I have one question that from the audience that Dr. Wang, you think you're able to share this PPT with the public, with the audience?
Yes, it's public. It's cleared by the FDA, yes.
So everyone listen to that. If you need the PPT, you can either send the email to the organizer, Dr. Chen Jiayu, and she will try to contact you. So Jiayi, I pass this over to Jiayi, and Jiayi have some, I'm sure have some more question about the treatment of COVID-19. We focus on the treatment of COVID-19 rather than other issue. And to Dr. Heston.
Thank you, Dr. Liu. I'm really impressed by your answers, Dr. Haseltine. So I noticed you published an article on May 19th. You have some criticism there was not sufficient data in Moderna's release. You said it's was premature and not impressive, and it was opaque on their results. And maybe the market had applauded their results more than they should have done. So now 2 months has passed. Have you changed your mind, or have you changed your opinion on their progress?
Let me first say, I'm reading a private note to me that suggests that I should apologize to Dr. Wang, and I'm happy to apologize to you for mistaking your presentation. If you were important in reversing the EUA on hydroxychloroquine, I certainly think that was a very good thing to have done. And perhaps my attention wandered before you got to your main point. So I apologize for that. And Dr. Peck, thank you for bringing that comment to my attention. Unfortunately, we still haven't seen the Moderna data. And they promised to have it forthcoming in weeks. It's been well over weeks. They have gone on to make other announcements that are equally unsupported, but they are not alone. You are hearing the same kinds of things from other groups, that they're meeting with various kinds of success, yet they're not publishing their data. The most recent one was again with remdesivir. It took them a long time to publish their results. And when they did, they were not particularly impressive. It received EUA, emergency use authorization. It is now widely used. In fact, people are going crazy trying to get it for a drug you really don't know whether it works. And now they've just announced that it helps people with terminal disease, but we can't see that data either. I think that this trend to help to try to get the data out, we understand you need data out. That's what all the journals are allowing you to do. They're allowing you to put your preprints out so you can have the data without all that delay that's normally involved with the review and publication process. That, I think, is the minimum of what you should be able to do. So you can at least see the data that supports the claim. And without that, we are at sea. Nobody can know what is going on. You need to see the data. So I don't think I've modified my opinion at all of what I said about the Moderna release. In fact, the fact that we don't see the data, the fact we have another opaque announcement for them that they've had to redo and rewrite their protocol is puzzling. In what respect did they have to rewrite it? Was the requirement to show that it actually reduced disease new to them? We don't know what it was. So I think that when people, and this is really important for public policy, people are hoping for a vaccine. Think of all the people who are thinking their children back to school. Will there be a vaccine this year or next year? This is people's lives they're playing with. At the same time, you could, whether it's true or not, make a reasonable assumption that they're making a lot of money off these announcements. All you do is watch their stock price and watch their internal sales. You could make that assumption. I'm not saying that you necessarily have to, but you could. And so you're in very dangerous territory with releasing public statements without the data that allows anybody to look at it.
Thank you so much. And it's also the similar situation to Oxford vaccine.
Oxford's a little better because they actually say on their site, don't listen to anything that people say. It's actually on their site. It says, only look at what's published here. Okay, that's on their site. So, and that went up before the very first announcement of their monkey studies. So at least they're a little better. But I would say for all of the vaccines, whether they're done in the United States, Russia, India, Singapore, we need to see the data. And I think that the analogy I've given, it's like a CFO of a company says, we've had great results, but I'm not going to show them to you. Who would believe that? Who would invest in that kind of company? And what would the SEC as a regulatory agency do? They would investigate and punish a company that did that. So I think it's not proper, especially when so many people's lives are at stake.
Yeah, and I have a following question about Moderna's products. So what does Moderna need to do for phase 2 before they can get into a phase 3 trial to determine the optimal dose?
I don't want to comment on that. I'd leave it up to the FDA.
Okay. And so there are different types of strains of vaccine have been found in Europe, in US, and in China. Do you think there are different types of vaccines need to be discovered for different?
What you're saying is there are different strains of virus.
Yes.
I don't, as far as, I've been looking at this question with the people who are doing the most advanced biochemical work, trying to understand what these implications are. And the most widespread virus is one which has a single mutation, which tightens up the association between the two exterior parts of the spike protein. And those virus particles have more functional spike and are up to 10 times more infectious in a tissue culture experiment. And the fact that combined with the fact that they spread more widely suggests that the virus that's currently spreading around the world is more infectious than the virus that originally came from Wuhan. And there's an additional study of China in Chicago, which has a virus that came very early on from Wuhan, very directly from Wuhan, that has not taken over. The other viruses from the West Coast and the European virus have swamped it. So there's a lot of epidemiological as well as now biochemical support for the fact that there is at least one important variant. Now, what is good about that is there's no observable pathology that differs as far as any doctor has seen that I know of. There may turn out to be. And secondly, it looks like it hasn't changed its neutralization. These coronaviruses aren't like the flu. They don't change. They have a nastier habit. They can come back and get you when they're not in disguise. At least flu has to disguise itself to get you. These viruses, I call them get it and forget it viruses. For some reason, your immune system seems to forget you were ever infected. And you can be infected by the same coronavirus. I think it looks like at this point, SARS-2 looks like those other viruses because in a series of experiments, people are watching antibody levels fade over time. And we have to watch that very carefully. But I think that at this point, a simple answer to your question, it doesn't look like at this early stage it's going to be like a flu that we're going to require multiple vaccines. However, unlike the flu, it's not clear that the vaccines are going to be as effective. And even for flu, they're not completely effective in stopping infection.
So why is it so difficult to defeat the virus when human beings already beat the bacteria and the fungi and the other things?
We have very good antiviral drugs for HIV, getting better all the time. This new drug from Gilead, GS-6207, is an amazing drug. Cabotegravir by VIF, VIIF, is an amazing drug. We're getting really, really good at that. We have drugs that cure hepatitis C. Egypt just eliminated hepatitis C from its entire country by testing everybody over 12 and giving them free treatment. They don't have hepatitis C in Egypt anymore, the most highly infected population. We have good drugs and have for a long time against the herpes viruses. There are some viruses where we haven't put enough work into. We should have put a lot more work into the coronaviruses because of SARS and MERS. We can, I believe, have very powerful, effective drugs, not just monoclonal antibodies, but I think we can do better with this virus, with drugs, than we can with HIV by far. I don't think it's going to be a big issue. I think within 2 years from now, we'll have a series of approved drugs, which, as our host said, should be used in combination, because we know, for example, even with the latest drugs that are being developed against the protease, in one of the early trials, already somebody was showing resistance. So we know these viruses get resistant. You have to use multiple combinations of drugs for this as well as others, but I'm pretty sure we're going to be successful. But somebody asked a question, which I want to get back to. They said, how can we stop the infection without a drug or vaccine? Ask the Chinese, ask the Taiwanese, ask the South Koreans, ask the Australians and the New Zealanders. They've done it. You do it through very rigorous public health measures. We humans can beat this virus if we're organized properly. We don't need a drug or a vaccine. We can do it through behavior change.
Meantime, could you please introduce Access Health International? Why?
Well, that's very nice of you to ask. I'll do two things, but thank you. First, let me say that I've written a book for parents and children. It's available on Amazon for 99 cents. And it's a new kind of book because it's a living book and it's an ebook and it updates automatically. So you buy it once and you have a code that lets you read all the subsequent editions. And even after 3 weeks, we've just finished this, almost finished the second edition, because things change very fast. Let me give you an example. When we wrote it 6 weeks ago, the consensus was that a pregnant woman had no additional risk. Today, we believe that pregnant women do have an additional risk and have the possibility that they will become sicker than most people just because they're pregnant. And there's a possibility that their infants may be infected intrauterine. Now, whether that's going to affect them adversely, we don't know yet. So all of that is new. So that changes. And Access Health is a global think tank that works on health systems. We work in China, mainland China. We have offices in Beijing and Shanghai. We have offices in Singapore and Manila and in New York. We've been very active in Scandinavia as well. And what we do there, and of course, all throughout India. We have offices in Hyderabad and Delhi. And what we do is we work on health finance and health policy. After many, many years of work in research and drug development, I've come to understand that no matter what we do, if people can't afford to buy the drugs and if the health systems aren't organized properly, it doesn't make any difference what we can do. So in order for us to be able to make people healthy, we have to have healthy systems. Government has to work right. And so we're privileged to be able to advise governments and the private sector to work on health policy and health finance.
Yeah, I was firstly impressed by your mission, life mission, a life to dedicate to devoting to improving human health on every scale. Yeah.
Well, I think everybody on this conference call has the same motivation. I think that's what we're all here for.
Thank you.
Thank you so much.
So I know we are close to the time, but if both of you are not so busy on the weekend, if the audience still have to be patient, I could extend it to this by our team decision for about 10 minutes. And then because we have a few questions from the audience, and I would like both of you to address it. Some of the questions specifically to one of you and the others. So Dr. Yaning Wang, there's a question about, we talk about a combination. We know there's a lot of clinical trial both everywhere, okay, in the worldwide. But as the FDA, how many cases are you handling? How many cases have you approved? Do you have that kind of number, like a combination of therapy against COVID-19? We only focus on COVID-19 at this stage. And combinational case, any individual single new drug. You don't need to reveal confidential information, and the audience want to grab available information. Are you able to address that? The second question is for Trevor, Dr. Hasselton. That is your opinion. I will let Dr. Wang answer first, is because it's relatively straightforward. The second question is more public health question. Should children return to the school in the state? Okay, Dr. Wang, you go ahead.
Well, my short answer is very simple. Some of the information I cannot share, or the only information I can share is the public information. So if you look, in the news, I think at least the Regeneron trial that's ongoing, and they just started the phase 3. That's a combination of 2 antibodies. There are trials or drugs like that, but I cannot share, how many are pending within FDA. So that's it. Thank you.
Thank you.
With respect to the school question, it's a question that's on every parent's mind around the world. And the answer is not a simple answer. I would say the first thing is an estimation of your risk. In the United States or wherever you may be, I would look at your postal code and try to understand how many people yesterday and in the previous week were infected in your postal code or in your local region. That will give you the best estimate of how many people are walking around with infections. So multiply that by 10 roughly, because people are infectious for a period of time, and you'll have an idea of how many people in your community. If it's 0, then in your group, it's 0. Then you have to consider how many people come into the school from other areas. So you have to consider a wider network, the janitors, the teaching staff, the bus drivers. What is the catchment area of infection that you operate in? That's the first thing. So I call that situational awareness. The second risk assessment you must make is my child at high risk? Does he have diabetes? Is he obese? Does he have serious asthma? All of those are not good for your kid. Or if he does bring it home, do I have elder parents? Do I have other people? Am I obese? Is my husband diabetic? Those are serious issues too. So you have to make a risk assessment, which is very difficult for people to do. They need help. We need very clear guidance for that. Then you have to look very critically at what the school is doing. How are they planning to protect your child? And the level at which you have to do this depends overall on the level of virus in your community. Let me say that clearly. In China, it's one situation. In Houston, Texas, and Miami, it's different. New York is different. North Dakota is different. Sweden is different from Southern Italy. You have to be, this is local. You're talking about sending your kids to school. And that's a very local activity. And so then you have to look very carefully at what the school is doing. Are they doing team sports? Not a good idea if you're doing contact sports. Are they separating your kids? Are they wearing masks? Are they doing face shields? What exactly are they doing? And what are their backup plans if somebody's infected? All those things, unfortunately, parents are now having to do. Then there's other serious questions. How well will my kid be socialized? How well will my kid, you can say I'm thinking about this. I'm actually thinking about writing another book right now on this topic. Because it's such a critical for our future. You know, it's for the future of psychology, the future of the health. It's really important what we're doing, how we get this right. Is virtual learning really learning for all kids? Those are questions. And now the question is, what are the regulations for homeschooling for those people who decide it's too dangerous to send your kid to school and they would rather have them homeschooled than, and there is a pathway in the US anyway for homeschooling. All these are really big questions. And I would say, just to make a simple answer, it's a very local issue that depends on what's happening. And one other point, what you decide for children K through 6th grade may be very different from what you decide for your high school students. High school students are more active, more susceptible, and they are going to be closer because they'll have sex. That's something that people don't like to talk about, but it is a fact of life that we in the HIV world had to contend with very early on, and nobody is talking about it for this disease. Young people need personal intimacy, and personal intimacy is a high risk. With HIV, you had to be very intimate before you contracted it. With this disease, you only have to cuddle. So it's a more serious issue. And I think we have to start educating everybody to know your partner. With that, maybe the biggest hope we have in that respect will be a rapid antigen test. The PCR tests are taking so long and are too expensive. I think we very badly need, and I hope the FDA is on this too, approval of a very rapid, specific antigen test, which means in 5 minutes you can know who you are with and whether they're actively at that moment infected from a saliva sample. Let's hope we have such a one soon.
Thank you, Dr. Hazelton. I have a few more questions from the audience, and this is a specific direct to Dr. Hazelton. One of the questions is that early on you mentioned that with the drug, the vaccine, maybe not something that we should rely on. We should rely on something else. But you also outlined a little bit, and from the audience, seems like they didn't get the detail. I should respect our audience because we have a different, we have lay people who not work on medicine, who not work on health science, and work in different aspects. So the question is that without rely on drug and vaccine, how can you give a little bit more detail, like a 1, 2, 3, 4 to our audience, to our public health, and to prevent, to overcome this COVID-19?
I think what you can look at is China is the case in point. Taiwan is the case in point. South Korea are case in point.
Dr. Hazelton,
let me be specific, okay? I'll get to specific.
A lot of people do not read news about us in the state. They don't necessarily.
That's unfortunate. If you want to know what happens, they have to know what happened.
They don't know anything about China. So
so what they did is, the first thing they did is they said everybody must stay. Let's take Wuhan, a big city. Everybody has to stay at home. And I was in Wuhan in November, and I was in daily touch with people in Wuhan during this whole crisis. And they said, everybody stay at home. And they were very strict about it. Nobody can go out. You can't leave your building. You need a pass to go out. That's the first thing they did. Second thing they did is they vigorously contact traced right away for the whole country. I'll give you one. Let me give you an experience. A friend of mine. I published it. I did a deep interview. He and his wife flew from Frankfurt to Shanghai in February. One person on the plane was infected. Without a single test, they contacted everybody on the plane and put them into a single hotel room for 14 days from the time that plane landed. And in that hotel room, they did not interact with another human being who wasn't in a hazmat outfit. They weren't even in the same hotel room. There were different hotel rooms. They had to disinfect their feces and their urine, something we don't even think about. And they were delivered food by a knock at the door by a hazmat-clad person. And he left, and then they opened it and they got their food. That is control. And they reported their temperature twice a day to the local health authorities. When they left, they got on their phone. They moved from orange to green. They could then go out and they could go to their own apartment and they could walk around to get food. But that is about all they could do. It was very strict control. They took a city which had thousands of infections a day and reduced it to 0 without necessarily testing, without a drug, and without a vaccine. And extend that, they stopped people traveling. Also, from one city to another. I had another person who left from Taiwan, went to Beijing, 14 days in her apartment. Somebody went from Taizhen, a coastal city near Beijing, to Beijing, 14 days in her apartment without going out. And there's a code. And if you get stopped without your code, you are in deep, deep trouble. wearing universal masks, total information about what you as a citizen have to do. That is an example of how you control this epidemic and drive it to zero. And eventually, if it does pop up, because it will, you do the same thing. You talk about testing. When a new series of cases popped up in Beijing and another series in Wuhan, they tested in Wuhan about 10 million people in 6 days for PCR. Same thing in Beijing, they tested millions of people within a week. In the northern cities, when there was a small cluster that came from people who came in from China, Chinese citizens returning, came in to some of the northern cities, they closed those entire cities until the infection had disappeared. That's how you do it. We don't do anything even remotely close to that. If you don't want to do that, you do what the Europeans do. You say, everybody stay home, and you enforce it. If somebody is out without a pass that says they can go out, they get huge fines, and if they do it repeatedly, they go to jail. That's how you do it. And everybody begins to agree. Now, as they open up, you can see that they're not as efficient as the Chinese, as I mentioned. If you were to translate the level of infection in Europe on a per population basis to China, China would be having several thousand infections a day. They're not. They are much stricter than the Europeans. Do the Europeans run a risk of having the whole thing blow up again? Absolutely they do, because they have not been rigorous enough in enforcement using the tools we have. The greatest tragedy in this is that very few people in the United States and around the world needed to have died from this disease if we had heeded what we observed from East Asia. Now, East Asia had an advantage. They had SARS. They knew that it would have an economic toll and a human toll, and so they acted appropriately. In fact, South Korea had even done a SARS exercise in early December before they knew that this was coming. So there was a mental preparation. The people who are now leading China have stamped into their memory that they were effective and promoted because they helped control SARS in their domain. That's one of the criteria for promotion to where they are today. And so they did have an advantage, but we had the advantage of seeing what worked over there. It isn't, maybe the average American didn't know what was happening there, but I assure you our CDC and our FDA sure as heck knew what was happening over there. I can't hear you.
Yeah. Thank you, Dr. Haselton. And to summarize what you just said is the best way to prevent this is to do community, society, government engaged isolation of a human being. That's what I would summarize based on what you said. If we can do best on this part, we will better prevent the COVID-19 pandemic. So another specific question for you is does the allergic asthma have the same risk as the general asthma? Do you have any answer on that specific question?
I don't have an answer on that. Maybe Dr. Wang does or someone else. I don't have an answer.
Dr. Wang, you have it. I think this is unfortunately we don't have a physician online here. If we have a physician online here, you're welcome to address this question. And Jiayu, you have a more question to Dr. Haselton or Dr. Wang?
Yes, I have millions of questions, but we almost run out of time.
Thank you. I want to thank the organizers, and again, I want to apologize to the good doctor.
No, let's. Okay, go ahead.
Let me say a few words. No, you don't have to apologize. Probably I did not get to the bottom line very clearly from the very beginning, and the talk went too long, so probably you lost patience. Did not even pay attention to my conclusions. That's totally understandable. Again, I apologize too for going over the time too much. And thank you for staying, beyond the planned time. And I guess it's my honor to give this talk together with Dr. Haselton. Before this event, I didn't know him, but after I read, his achievement over the years, I was really impressed. That's why I feel it's really an honor to do this together with you.
Thank you. Thank you very much, everybody.
So because the time, we already over time, run out of time for 15 minutes. So even though there are still a long list of questions coming along. So in the future, if you have more questions toward Dr. Hasseltine, Dr. Wang, good, I'm sure you can find their email, their website. You can contact them directly. And as a non-biologist, as a non-clinical doctor in this area, I'm a biochemist. So I really learned a lot by talking to them and listening to them. And I particularly appreciate the organization that organized this event, the Chinese Association for Science Technology. Even though this is a historic society, I feel very solid. It's the first time involved in this activity. And I appreciate Jia Yi to invite me over and to learn this and to experience this. Particularly the first time from online, I learned both of your study and your great work. However, the first time we have direct interaction, from my personal point of view, I really appreciate this event, even though it's a short. I wish this can go to two more hours. I know audience, I have to apologize to audience and that we cannot continue because this is already almost 20 minutes beyond our original caster, and Dr. Hasseltine, Dr. Wang give up their weekend to share with us their very informative, valuable opinion as well as the information. And I actually appreciate Dr. Wang's detailed presentation through the molecular detail, to explain why FDA revoked that decision, which is great. And also appreciate Dr. Hasseltine's very sharp point of view about how we should overcome COVID-19. I know when you say drug and vaccine is not necessary, you don't mean that. You actually, drug and vaccine are necessary, but that's the only way we can eventually overcome this. And even though we still do not have a great vaccine for HIV, great vaccine for flu, we still work on it. But I let this last part to the Jia Yi. Jia Yi, I want to be grateful to you and the rest of the team. There's a lot of people behind the scene doing this, and I just want to thank you. Thank the audience to be patient with us, okay? And Jia Yi, it's your turn.
Thank you, Dr. Liu. Well, this virus has a very dark side in all the pain and suffering. And we are, as an association, we trying to give our audience hope through this pain and suffering, and eventually we will bring the world closer together. And Dr. Hasseltine, Dr. Wang, thank you so much for an enormous number of insights packed into the last 90 minutes, more than 90 minutes. Appreciate very much on behalf of the CAST Greater New York chapter, you joining us. Also, I want to thank President Tao, Karen, Kevin, our volunteer team for being with us. And thanks again to Dr. Liu, also joining me for the fireside chat.
So goodbye, be safe, everyone.
Thank you.
Hey, Dr. Hasseltine, Dr. Wang, and
Thank you.
hang around when we still have a pandemic going on in the country. They're hanging around. Thank you, Jia Yi and the team, Dr. Tao and Xiao Tao and the team. Thank you. Thank you, everyone. Have a nice weekend.
Yeah, have a nice weekend. Thank you, guys. Appreciate it.
Thank you. And for audience, if you still need the slides, you can still send your email privately to me, CAST-GNY Karen, and I will collect all your emails and then send out the slides.
You can, Claudia, can you just post your email address on the you can wait it before you terminate this event.